and how it can be used to study biological samples. In a physical lab, this room would be dark to protect the sample and get better quality images.
This simulation uses voice.
Using the lab path. Perfect. Then let's get started. Find a glove box and click on it. One of the researchers has already prepared a chicken intestine sample for microscopy. The microscope parts are on the workbench. Assemble the microscope to match the image on the screen. Interaction is not possible with the item in hand. Interaction is not possible with the item in hand. Interaction is not possible with the item in hand. The ocular or eyepiece lenses add a 10 times magnification. The microscope camera makes sure that the image is displayed on the screen next to the microscope. The objectives are the microscope's lenses. They magnify the sample. The microscope stage is where you place the specimen. In our case, the slide that your colleague has prepared. This microscope, nice job! Check out the big image on the wall screen to see some components that are inside the microscope. Remember that you can also click View Theory to learn more about the microscope. What is the purpose of an emission filter in the fluorescence microscope? Option A. Only the excitation wavelength reached the eyepiece and camera. Option B. Only the excitation wavelength reaches the sample. Option C. Only the emitted wavelength reaches the sample. Option D. Only the emitted wavelength reaches the eyepiece and camera. This is not quite right. Please try. What is the purpose of an emission filter in the fluorescence microscope? Excellent thinking. The emission filter only lets through the wavelength of fluorescence is generated when fluorescent molecules that are brought to a higher energy state fall back to the ground state. The molecules lose some of this energy, for example through heat, before falling completely back to the ground state through fluorescence. The lower energy of the emitted light means this light has a longer wavelength. Since we are using the 100 times oil objective, please add immersion oil to the objective. Now place the slide on the stage. Well done. The DNA was stained with DAPI, a fluorescent dye that binds to the DNA. This allows you to visualize the nucleus. Here, DAPI is shown in cyan. The microscope is explore the magenta and yellow channels. There is no fluorescent dye for the viral protein, but luckily, there is an antibody that binds to it. Actin is a part of the cytoskeleton and abundantly expressed in all cells. In this case, we are using it. As you have seen, there are different ways of labeling a molecule of interest. You can add a fluorescent dye or use an antibody with a fluorescent molecule attached to it. You could also use gene editing when you want to label multiple structures in the same sample, you can combine different types of fluorophores. In our experiment, for example, we are using 2 different fluorescent dyes and an antibody. Which of these can be an advantage of fluorescence microscopy over light microscopy? Option A, study cell processes in living cells. Option Smart. Compared to light microscopy, with fluorescence microscopy, it is easier to explore the sample in all 3 channels. When you are done, click the overlay button to merge all 3. Each fluorophore has its own excitation and emission spectrum. When using multiple fluorophores in the same sample, as we are doing here, we need to ensure the emission spectra do not. If you want to image multiple structures using fluorescence microscopy, which of these considerations? Indeed. The emission wavelengths should overlap as little as possible to avoid the signal from one of the molecules bleeding through into one of the other channels. Now look at the overlay image again. Remember that you can see the cells outlined in yellow with the nucleus in cyan and that the virus is labeled in magenta. Does the virus infect all the cells in the chicken intestine to the same extent? Great job! You not only know how a microscope works, but you can also interpret a micrograph. The virus, which is labeled in magenta, is present in the empty lumen and in a few cells. This is the strongest magnification this microscope can achieve. Like the light microscope, the resolution of the fluorescence microscope is limited by the wavelength of the light. In this case, viruses are very small. To learn more about the localization of the virus inside the cells, you could use electron microscopy. The wavelength of electrons can be up to 100,000 times shorter than that of light, which means that the resolution is many times higher. In this EM image that I took earlier, you can clearly see the microvilli as a brush on the cell surface on the right-hand side, thanks to the increased resolution. We were unable to see these tiny extensions with fluorescence microscopy. Electron microscopy images are a gradient of the number of electrons that pass through the sample. Advanced image processing techniques in combination with specialized microscopes have been developed in recent years to overcome the resolution limit in fluorescence microscopy. Using these super-resolution techniques allows scientists to image samples with a resolution. Some are even performing electron microscopy and super-resolution microscopy on the same sample to combine the localization information of fluorescence microscopy with the ultrastructural information of each. Let me ask you one last question. Do you feel